Welcome to section 7.2, Meiosis. In this section, we'll focus on how meiosis creates gametes with half the number of chromosomes, and why this process is essential for genetic diversity and evolution. By the end of this section, you should be able to describe the behavior of chromosomes during meiosis, explain the key cellular events that occur, compare meiosis with mitosis, and explain how crossing over and independent assortment generate genetic variation among gametes. Meiosis is a type of cell division that reduces the chromosome number by half. One diploid parent cell divides to form 4 haploid gametes. Unlike mitosis, which produces identical daughter cells, meiosis produces gametes that are all genetically unique. This uniqueness is what makes sexual reproduction such a powerful driver of diversity. Let's define ploidy. Ploidy refers to the number of complete sets of chromosomes in a cell. Diploid cells, written as 2N, contain 2 sets of chromosomes, one from each parent. Haploid cells, written as N, contain just one set. Meiosis reduces the chromosome number from diploid to haploid, ensuring that when gametes combine at fertilization, the resulting zygote has the correct diploid number again. Mitosis and meiosis are similar in that they both involve chromosome duplication and division, but their outcomes are very different. Mitosis creates 2 genetically identical cells used for growth and repair. Meiosis creates 4 genetically unique gametes with half the original chromosome number. The unique steps of meiosis, crossing over and random assortment, are what generate variation. Meiosis occurs in 2 rounds of division. In meiosis 1, homologous chromosomes pair up, exchange genetic material, and then separate into different cells, cutting the chromosome number in half. In meiosis 2, the sister chromatids of each chromosome separate, much like in mitosis. By the end, 4 haploid gametes are formed, each with a different genetic makeup. Meiosis generates variation through 2 main mechanisms. First, crossing over occurs in prophase 1, when homologous chromosomes exchange segments of DNA, creating new allele combinations. Second, independent assortment occurs in metaphase 1, when homologous chromosome pairs line up randomly. This random arrangement ensures that each gamete receives a different combination of maternal and paternal chromosomes. The outcome of meiosis is genetic diversity. This diversity is the raw material for evolution, allowing populations to adapt and change over generations. For example, the adaptive radiation of the Hawaiian honeycreeper, with its wide variety of beak shapes and feeding strategies, reflect the power of variation created by meiosis and sexual reproduction. We can see the results of meiosis all around us. In animals, it explains different fur patterns. In humans, it's why siblings have distinct traits even though they share the same parents. In plants, it accounts for variations like flower colors. These everyday examples illustrate how meiosis drives the diversity of life. Let's bring these key ideas together. Meiosis is essential because it reduces chromosome number by half, making haploid gametes that can combine at fertilization. The two divisions, meiosis 1 and 2, ensure that homologous chromosomes and then sister chromatids are properly separated. Along the way, crossing over and independent assortment shuffle genetic information, guaranteeing that every gamete is unique. This genetic variation is what drives diversity within populations, fueling adaptation and evolution.